Okay, so we're running a bit late, and it's Friday afternoon, and I'm sort of under the weather and probably spend about half my time coughing. So I'm going to keep it short and light and basically just show you some pictures. Okay, so every first-year medical student studies anatomy, basically the structure of the human body. And that's important because our understanding of how the human body functions and how to treat disease is based largely on understanding of that structure. So anatomy is usually viewed as very coarse level structure. But we can go down to somewhat finer scales, sort of to tissues and cells, by looking through microscopes. But of course, there's a lot happening at scales much finer than that, basically all the way down to individual atoms. So it's possible using experimental techniques like crystallography, X-ray crystallography, to deduce the atomic level structures of the molecules that make us up. This is a protein, an adrenaline receptor. So one can get that structure experimentally, and then using computational methods, basically physics-based simulations, one can figure out how these proteins move at an atomic level. So I've spent much of the last 10 years working on these kinds of physics-based simulations, and using them, for example, to understand how drugs bind to their targets, where they bind, how they exert their effect, like what they actually do to the protein target, or to understand how proteins function. So here's a rather large protein that undergoes a large structural change as it functions, and that's what allows it to release a bound molecule. Okay, it's kind of a key event in molecular signaling, and it's important from a drug development perspective. So I basically have two goals over the coming years. One is to take this technology further to the point that it can really make more of an impact on drug development. And I'm working with a number of drug companies to that end. The second goal is perhaps a slightly more difficult one. So I've talked about sort of a very high-level structure, like as we think about it in anatomy or at a tissue level. And I've talked about very fine-level structure, where we're thinking about individual atoms. It turns out there's a regime in between that's actually harder to get at. So between sort of anywhere from large groups of proteins coming together to form larger machines, like some of these, to the organization of the whole cell. There's still a great deal of structure, but it's harder to study. It's harder to study because it's hard to observe with kind of the macro scale observation techniques, looking by eye or under a microscope. And it's also harder to get at with techniques like crystallography because everything is sort of in flux at this scale. And it's also much less clear how to study it computationally. In particular, it's at these scales. Actually, let me just show you a nice little movie. This is an artist rendition done with kind of software that's used to make Hollywood movies. Just shows you a little bit of a cell in action. So the question is how to really get at what's happening at these scales. It's not easy computationally because we can't just use physics-based simulation. At least I believe it's hard to sort of get this all kind of building from basic physical principles up, in part because there's so much there that we don't even know what it is. We don't even know what's present. So how can we get at it? We need to use a more data-driven approach. I believe in concert with simulation and other physics-based approaches. And fortunately, there is a lot of data beginning to be available experimentally on what's going on. For example, one can use single molecule fluorescence microscopy to track individual molecules. But that experimental data gives you only a very partial picture. It allows you to see only certain things at a time. I'd like to use computational methods to combine it all together into models that are A, comprehensive, and B, predictive. Predictive in the sense that they allow you to predict what will happen if you perturb the system, for example, by adding a drug. So I will wrap up there. Here's my email address in case you have questions afterwards.
So I guess the problem with a data-based approach is that you sort of only model things that have already happened, say. So if you want to say study what happens if you change the system, how do you think that's going to, like, what would be your idea for how to approach that?
So I think you need to use a combination of two ideas. One is a sort of statistical modeling or machine learning idea, where you think of what you've seen as examples of some broader statistical pattern and then draw inferences for new cases. And the other is simulation. Not the same kinds of simulation that I was showing you previously, but some kind of simulation that really has predictive power. That's sort of my high-level answer.
Okay.
Yeah.
Just curious, were those ion channels that you're trying to predict how chaperone things actually go on allow your ions to go through your molecule?
Well, I didn't. I have worked on ion channels. I didn't show any pictures of ion channels. I showed pictures of G protein coupled receptors and G proteins and chaperones. Okay.
Yeah, well, thanks so much, Ron, for coming, despite the fact that you are not feeling so well. So really appreciate that.